from Microbe TV. This is TWiP, This Week in Parasitism, episode 218, recorded on June 22nd, 2023. I'm Vincent Racaniello, and joining me today here at the incubator in New York, Daniel Griffin.
Hello, everyone.
Good
Physically here at the incubator.
Yeah, you should come more often. You should come once a month. That would be a good goal, right?
That would be an excellent goal.
Right. Also joining us from Glasgow, Scotland, Christina Nowla.
Hello, everybody. I should come to the incubator one day. I'm quite keen to visit. Looking for excuses.
You should. Well, aren't you coming to ASTMH?
I am, but isn't that in Chicago?
Yeah, but you could.
I could stop over.
Can you fly to Chicago? Can you fly to Chicago from Glasgow directly?
Not from Glasgow, but from Dublin.
So could you go to Glasgow to New York?
I probably could.
Not,
yeah,
right?
I'm sure I could.
Edinburgh has a flight actually that I take.
All right.
So on the way back, or on the way there, you can stop in New York because you're going to, might as well. You're on the continent, as we say.
I know. I'm always looking for excuses to travel.
Oh, you like traveling?
Yeah, well, I don't like the actual traveling, but I like to be somewhere else.
Oh, I see.
So I'm really looking forward to
other than where you are.
Yeah. No, I just don't like the airplanes and stuff like that. But,
once you get there,
once I get there, it's fine.
All right. We have two cases. Boy, Daniel, you really outdid yourself. I hope you don't have three next time.
We're going to keep, pretty soon we'll be up to 12. But okay, let us, as I like to say, let us remind those tuning back in. And for those showing up for the first time, let me relate the story. So we have a man in his 60s who provides IT support for a bank, all done via Zoom and remote work, who is referred to me. And he reports having issues after a COVID diagnosis, September 5th, 2022. He had 4 vaccine doses, and with diagnosis was treated with Paxlovid. He feels better. Then about 3 weeks, about week 3, wakes up with URI symptoms for about 1 week. And by the 28th was improving. He had cold agglutinins detected, referred to ophtho by his primary with some eye discomfort, and noted to have elevated intraocular pressure. Sleep and fatigue were a problem, slowly improving. Cognitive issues noted, but improving. Now, he reports an issue a few years prior to this current problem where he developed fever, a sore throat, tender lymph nodes in the neck, in the front and in the back. This resolved after about a week and was then followed by months of fatigue, sleep disturbances, not feeling well. He reports no specific dietary preferences and reports no cat exposures. He saw several physicians and one did a number of blood tests. He actually had a patient today who grew up in Puerto Rico, but reported she had never been bitten by a mosquito in her entire life.
Oh, I'm sure she was confident about that. Yes.
I try to maintain that, that physician demeanor. And I said, really? You mean to tell me that? Okay. A man. Okay. So that's our first one. As I said, he saw, so we'll go back to that one. Sort of got off there. So as we mentioned, he had this issue before, and the no cat exposures. He saw several physicians, and one did a number of blood tests that we will discuss. But we also had a second case because that wasn't enough. A man in his late 30s presented to an ER at an outside hospital prior to ultimately being transferred to an academic center in New York City. He reported onset of left arm weakness that prompted him to come to the emergency room. He reported on pointed questioning that he had COVID about 1 month prior and felt he had fully recovered. He did have a headache that preceded the onset of weakness. The outside hospital triggered their stroke protocol and performed a head CT, which revealed a hypodense lesion on the right side of the brain not consistent with a stroke. A bit more history is obtained, some further testing is done, and based on this, the patient is transferred to our academic hospital on some sort of therapy.
Okay, Christina, can you take the first one?
I can. So Hawken writes, hello again from sunny Athens, Georgia. 88 and sunny here. Given the questions about contact with cats or undercooked meat, my guess for this week's 2 cases are both Toxoplasma gondii. Given the cat questions for the first and the HIV positive status for the second patient, this was the parasite that came to mind first. Second to this was granulomatous amoebic encephalitis, GAE, from the amoeba A. castellani. Less likely, though, I settled for toxo. Sulfadiazine for treatment, I assume. Thanks again for another great episode. Hawken.
Oh, Athens, Georgia. I'm going there tomorrow.
Oh, fantastic.
For ASV. Very good. Daniel, you take the next 2, I guess.
Okay. Owen writes, dear TWiP team, I think both of these patients have quite different manifestations of toxoplasmosis. I think the use of both cases to illustrate the broad disease spectrum, very helpful. Hope you're all well. Justin writes, hello, TWiP presenters. It is 14C and rainy here in Ontario, Canada. A nice change from the smoky weather we have been having lately due to wildfires. My guess for the double case from TWiP 217 is toxoplasmosis due to the flu-like symptoms and eye disease of the older patients and the headache of the immunocompromised patients. Thank you for the lovely podcast.
So those wildfires, that's why I asked you about COVID. Could that lead, because people are saying that this could lead to more severe COVID?
Yeah, it is interesting because, we don't, as we talked about on our clinical update, we don't have great data specifically, but we do have a lot of data over time. What we used to see, out west, was an increase in the hospital admissions for respiratory infections. There was a nice study out of California. It was like a 2% per day increase for every day that the air quality was above a certain threshold. So certainly is a huge issue for people with pulmonary challenges.
Lauren writes, greetings to my esteemed teachers of the microbe TV universe. My diagnosis is T. gondii for both patients. I would have been clueless without the hint about the same diagnosis fitting both patients. I did not use ChatGPT, boring, but did go down quite a few search engine rabbit holes that finally led me to the fine NIH website. I learned much about toxoplasmosis, in particular how common the infection rate is, both worldwide and in the USA. Also about the reversible differentiation from the bradyzoite, chronic infection, usually suppressed by a healthy immune system, not really treatable, to the tachyzoite, active infection, new or reactivated. Drugs exist for treatment stages in the infected host. So I don't know if you guys remember, I kind of got off on ChatGPT last time, right? Saying people shouldn't. I got a lot of emails by mad people who said, Vincent, do you use a calculator to do your math? It's the same thing. I don't think it's quite the same because ChatGPT sometimes gives you nonsense, right?
Someone suggested to me I should use ChatGPT for marking. So it is tempting, but I wouldn't know how to go about it, but maybe something to think about for me.
I don't know. I just think for a clinical case, it's more fun to dive into the literature,
and
right?
also going down that rabbit hole on Google is quite nice.
Yeah, I think you'll learn a lot. Well, unfortunately, it's going from, maybe we're dating ourselves, but going from, when you used to read the newspaper and page through, now, unfortunately, we all live in this world where we sort of see stuff that confirms our bias because we sort of funnel our news. And one of the nice things about trying to figure out a case and doing a few searches, you read this, you read that, I think you can learn a tremendous amount. Yeah,
I don't have any opposition to ChatGPT. It's certainly good for some things, but I think something like this, you learn how to look stuff up, and that's what science is all about. It may change one day, but for now.
Well, I have an affection for ChatGPT because ChatGPT wrote a love poem to me the other evening, and so I was,
Really? Very nice.
It was very nice and very flattering.
I'm going to ask him to write me one.
All right, back to Lauren. The first gentleman, 60s, HIV negative, works in IT for a bank, got all the right prophylaxis and treatment for COVID, is suffering from a reactivation of a dormant infection with T. gondii, in addition to his PASC, or possibly creating all the symptoms that might otherwise be interpreted as PASC. Interesting. The initial infection was likely immediately prior to the incident, he reports, from several years prior, with symptoms of fever, sore throat, tender lymph nodes for a few weeks, and then ongoing sleep disturbance and fatigue for many months, which eventually went away. To be explicit, the symptoms went away, but T. gondii never goes away. It just lies in wait. Presumably, in his case, it insisted in the brain and CNS. I am fabricating a story to go along with this, with a guy in his late 50s who has heretofore led a structured life, deciding to do something a little bit out of the ordinary, like take up with a new partner who has cats, travel, go out to eat, and generally live it up in comparison to his previous habits. Maybe he just took up gardening. My fanciful story is just to explain a change of environment that exposed him to T. gondii when he previously had not been so exposed. Pardon my digression. The episode of COVID in September 2022 somehow woke up the dormant T. gondii, presumably because the immunological resources previously used to suppress it were distracted and spread thin by the coronavirus infection. Also, Daniel mentions in passing that this gentleman is a remote worker, providing IT support via Zoom and other platforms. Maybe he was given this assignment as a reasonable accommodation to an immune-compromised condition not otherwise mentioned. Or maybe he is immune-compromised, but without that being diagnosed, and his working remotely is just a coincidence. I hate to disregard this bit of personal information or any other in case it's relevant. I understand that those of us in our 60s do experience a decline in immune system vigor, so the age of the patient may be relevant. However, it momentarily escaped immune suppression. T. gondii took the opportunity to work on trying to complete its life cycle with quiescent bradyzoites, morphing into active tachyzoites, reproducing the symptoms of sleep disturbance and fatigue experienced in a previous illness. The eye was either newly infected or the reactivation of a previously dormant cyst created the symptoms of elevated intraocular pressure and vision abnormalities, and persistent fatigue resulted from the body's ongoing efforts to fight off the infection, diverting resources from the more desirable zest for life and energy for having fun of all kinds. A blood test for antibodies can distinguish between an active infection and a previous infection presently dormant. So his test could have confirmed an infection recently active. I wasn't sure what was meant by URI symptoms, but assuming it's upper respiratory infection, I don't know how to fit that into his case. Patient 2, the gentleman in his 30s, probably previously unaware of his HIV status, not under any sort of regular medical care. If so, he really had a lot to take in. COVID infection must have been the last straw for his immune system, and a previous latent T. gondii infection reactivated with a vengeance. Complications from T. gondii infections are very common and well known in persons living with HIV. Interferon gamma and its production by CD8 positive T cells are essential for controlling the proliferation of T. gondii tachyzoites. The high viral load relating to HIV, understand that the terminology is somewhat problematic, and extremely low T cell count would likely have led to the patient being started on antiretroviral therapy without delay, as Daniel mentioned, that the patient had already commenced some sort of therapy. Finding of a lesion in the right side of the brain upon CAT scan, inconsistent with stroke, but consistent with a common infection seen in HIV-positive persons, would have led to the referral of this patient to a preeminent academic center for the diagnosis and treatment of infectious diseases. Headache symptoms could be explained by the observed lesion, presumably reactivated, and the left arm weakness preceded by the headache could also be caused by the observed lesion in the right side of the brain. Wishing both patients good outcomes and glad they are in good hands. I never miss an episode, but often listen later, trying to find nooks and crannies of time while driving or performing other necessary but tedious chores. Best regards, Lauren. Very good, Lauren. I enjoyed that.
Me too. I think it's my turn, isn't
there you
it?
go,
Yeah,
Christina.
Kimona, right?
Yep, it is.
I was just trying to figure out if listening to TWiP was considered one of the other necessary but tedious chores. I hope not.
Oh, I don't think so. How can it possibly be a tedious chore? Okay, Kimona writes, greetings again from Vermont. A man in his 60s who has had no cat exposure is pure poppycock.
Very good.
That's a good point, actually. But perhaps I state this in an effort to make both these men's diagnoses conform to toxoplasmosis. It's like maneuvering that puzzle piece into a slot where you know it may not belong. Toxo is my best guess for these two men, presenting with CNS involvement and no mention of travel, hiking, lake swimming, poor sanitation, animals, or unusual diet, and residing in the northeastern US. This protozoan is ubiquitously found throughout the world. It can be acquired from cat feces exposure, the definitive host, but also from eating undercooked, contaminated pork, beef, or lamb, intermediate hosts, and can persist asymptomatically in a healthy person for long periods of time until perhaps the immune system is weakened. Patient 2 is HIV positive with a very low T cell count and susceptible to opportunistic infections or reactivation of a dormant state. The hypodense lesion on head CT can certainly represent CNS toxoplasmosis, which would reproduce encephalitis, headache, and then focal neurological deficits such as his left arm weakness. Patient 1 is perhaps not as straightforward. A recent COVID illness, cold agglutinins, and an eye problem. The cold agglutinins could indicate an autoimmune hemolytic anemia following his SARS-CoV-2 infection, leading to further immunodeficiency. His illness a few years prior could have been acute toxoplasmosis, which clinically can mimic mononucleosis. But even a true EBV infection can in rare cases take on a chronic active state and induce a progressive immunodeficiency involving both P and or T cells. Who knows? T. gondii may have lain dormant in this man since exposure in childhood, playing in the sandbox where a neighbor's cat would visit, and is now reactivated due to his weakened immune system. His eye ailment may be uveitis or retinochoroiditis, as seen in ocular toxoplasmosis. Diagnosis can be made by PCR or detecting IgG or IgM antibodies on serology, IgM being more indicative of an acute infection. Treatment of toxoplasmosis in immune-deficient patients uses a combination therapy with pyrimethamine and sulfadiazine for both neurologic and or ocular disease. In a healthy individual, treatment is often not required. However, if these two patients have disease caused by two different parasites, then I would also consider a tick-borne infection, such as babesiosis for patient 1. Babesia microti has become more prevalent in the Northeast and can exist in a carriage state for long periods and then undergo reactivation, similar to T. gondii. Several articles also describe the importance of CD4 T cells in the clearance of Babesia from within red blood cells. Not sure it would explain his eye ailment, though. As always, in eager anticipation and sincere appreciation, Kimona.
Retinochoroiditis.
Thank you.
Just rolls off the tongue, right?
It does.
Yeah, I have to apologize.
Daniel, you're next.
All right. Paul writes, hello from Petoskey, Michigan, 30 miles from the University of Michigan Biological Station, where I recently learned, I believe it's Dr. Despommier, spent educational time in the 1960s. These two cases are examples of toxoplasmosis in immunocompetent and immunocompromised hosts. Case 1, toxoplasmosis with ocular involvement. The case suggesting the diagnosis was likely IgM serology to indicate recent infection. Most cases resolve spontaneously in the immunocompetent. Treatment of a symptomatic case with ocular involvement would be pyrimethamine with sulfadiazine and leucovorin. Differential diagnosis of parasitic diseases with ocular involvement in the U.S. would include toxocariasis and acanthamoeba, which have clinical stories quite different from the case. Case 2, cerebral toxoplasmosis in a patient with HIV with CD4 count less than 50. Differential diagnosis is complicated, including TB brain abscess, cryptococcosis, amoebic brain abscess, neurocysticercosis, nocardiosis, invasive aspergillosis, and lymphoma or other malignancy. Diagnoses in HIV disease is complicated. Serology may be negative in the immunocompromised. Performing a spinal tap for cerebral spinal fluid for diagnostic studies in a patient with a brain mass risks the catastrophic complication of brain herniation. Brain biopsy is invasive and tricky and risky. Treating for 14 days with pyrimethamine, sulfadiazine, leucovorin, observing the response, and doing invasive testing if there is no response appears to be the safest strategy. Antiretroviral treatment should be started after initial antiparasitic treatment. Paul Blanchard.
Well, it was a thin month this
All
time.
right.
That's it for our guesses.
Oh my God. And these I thought were like, really, really easy.
Yeah, I think they're all good,
Yeah, I
right?
think maybe we're recording a little bit earlier than usual, so maybe a few diagnoses will flutter in over the next few days.
Could be. Maybe there's one here that on my
Yeah,
email.
well, while you check that, Christina, did you have any thoughts? What were you thinking?
Well, I thought those letters, I particularly liked Lauren's letter, and I thought, I would probably also diagnose these patients with toxoplasmosis. For the first one, I wasn't too sure whether it would be a new or a reactivation case, but he does seem to be, well, he probably is immune suppressed after COVID, and then also maybe in relation to his hemolytic anemia, as shown by the cold agglutinins. So, yeah, I think I'm fairly certain. I'm particularly certain for the second case. This sounds like a...
So you're going with toxoplasmosis.
I am going with toxoplasmosis.
It is the same diagnosis in both cases.
Yes, I am definitely going with toxoplasmosis. I'm just more certain with the second one as to how he developed symptoms, signs and symptoms. But I'm sure he will tell us more about your first patient.
Yeah, Vincent, what are you thinking?
Yeah, I think, well, this made perfect sense to be toxo because you would use it to illustrate the different presentations in a different host, right? An immunocompetent and an immunosuppressed host. I think that's a beautiful, as someone said, way of using case studies. And so the toxo was very clear in the second, less so in the first, but I thought it should be the same. So we're going to be toxo.
Yeah, those were sort of my goals. So yes, I'm going to say this was toxoplasmosis. And, start with the first case. I mentioned several physicians did a number of blood tests. And at that point, it was diagnosed as acute. It was actually acute toxoplasmosis in an immunocompetent individual, right? So he had come in. He had the fever. We talk about a mononucleosis type presentation, right? But not EBV, tender lymph nodes in the front and the back. And toxo is kind of classic for in the back as well. But the interesting thing, right? So he gets diagnosed. Initially, he's IgM positive, IgG negative, later becomes IgG positive. But he almost has a post-acute sequelae with the toxo for months. He is just fatigued, sleep disturbances, not doing well. So it's really interesting to me to have him now getting referred to me with post-acute sequelae of COVID with this previous story. And as we've talked a little bit, there's this issue. Is there some kind of reactivation of other pathogens that might be part of post-acute sequelae of COVID? Is there some sort of a genetic tendency, maybe something about how your immune system reacts where you're not sick just for a week or two, but it takes months before you start to improve? So I also wanted to put that in. But it's also nice to make the comparison between usually what is an acute self-limited, sometimes self-limited to a week or two or a few months, versus what we were seeing a reactivation for the gentleman in his 30s who at some point probably had a self-limited, but now that his immune status has diminished, he actually goes ahead. And in this case, this gentleman was treated, responded to treatment, reimaging showed that improvement. So in most cases, we can actually avoid any kind of invasive biopsy. We can just go ahead and treat.
So in both cases, will there be no more T. gondii present?
You never really clear it. That's the problem. Yeah. Some pathogens we control, but I don't think we ever sterilize the system.
So in case of immune disruptions, they could cause problems again in the future, right?
Yeah, so our goal here is to treat acutely,
Yep.
but also to restore the immune system.
So what was the treatment again?
So, it's changed a little bit over time. A lot of people historically would use a sulfa, so a sulfadiazine plus a
Okay.
pure methamine. There has been a little bit of a shift to using trimethoprim sulfa as our sulfur drug. Part of it is availability, part of it is cost.
And for the second patient, you also initiated antiretroviral therapy?
Exactly. And for the first patient, right? Well, oftentimes we won't even treat an acute. We'll just let it run its course. But yeah, we want to do a combination of antiparasitics and antiretroviral therapy.
I went to a talk at ASM on diagnostics in the setting of prescribing antiretrovirals for HIV. The idea that first you want to know the resistance profile of the isolate, right?
Mhm.
And second, the viral load in many cases is informative. And she said, person was from Hopkins, I think. She said, not everyone does viral loads.
We would hope they do. I think the standard is to know the CD4 cell level.
Right, for
It's
sure.
also standard not only to know the viral load, but actually to look at the genotype, which we now have a tremendous amount of data. So we could take that genotype and really understand what the phenotype, what the resistance patterns would be.
And they just to diverge a bit, she talked about other viruses, and herpes simplex was another where the mutations that confer resistance to acyclovir are all over the gene. It's incredible. They're terminators, right?
Yeah.
They could be anywhere.
Yeah.
Really cool. All right. Anything else, Daniel? Is that good?
I think that was good. And for the folks that wrote in, I think we had some really good responses this week.
All right. We had 8. We had 5.
Okay.
I believe everyone is eligible. I think Owen won a book, so I'm leaving Owen out. I don't know if I sent it to him, but certainly Owen. But now that I have a studio assistant, I'm going to have them send a bunch of books to Kara, and then she will send them out.
Okay. Sounds good.
All right. 1 to 5. What's our number? It is 3. Number 3 would be Lauren, the winner of a virtual book, right? Because I just don't send them out in time, but we will. So, Lauren, send your address to TWiP@microbe.tv. If you are living outside of the U.S., we will also need your telephone. All right, I have a paper for you today which caught my eye, published in the Proceedings of the National Academy of Sciences. Malaria parasite evades mosquito immunity by glutaminyl cyclase mediated posttranslational protein modification. So don't go away. It's not that bad. It's actually pretty cool and pretty straightforward.
It was a fantastic paper. I really liked it.
A lot of good biochemistry and genetics, as well as parasitology. So we have Surendra Kumar Kohli and Alvaro Molina Cruz and Tamasa Araki, and then we have Chris Johns as the last author. And these individuals are all over the place, and you have to go to the end of the paper to find out that they are from Leiden University Medical Center, the National Institutes of Health, the National Institutes of Infectious Diseases in Tokyo. What else do we have? We have the Imperial College London. We have another laboratory in Japan. Yeah, and the Netherlands, Leiden. What is glutaminyl cyclase? Well, it's an enzyme. We're going to call it QC because Q is the single letter code for glutamine, which is an amino acid. And this enzyme, QC, it modifies the N-terminal either glutamine or glutamic acid residue of a protein, and it makes it cyclized. It cyclizes it to what's called pyroglutamic acid. And this is a post-translational modification that is found in both eukaryotes and prokaryotes. There are 2 kinds of enzymes, mammalian and then the enzymes that are in bacteria, plants, and parasites. So looks like it arose at least twice in evolution. Mammals have two forms of the protein. They have one that's secreted, and then there's another that's in the Golgi complex. In mammals, it's implicated in stabilization of proteins like neuropeptides and cytokines. It's been associated with pathological processes such as amyloid diseases like Alzheimer's. It's been shown to modify the N-terminus of CD47, a protein important for myeloid cell immune evasion. We'll get back to that in a bit. But in parasites, nobody knows what these QCs do. Unknown. So that's what this paper is all about. They're going to look at what this does. Now, perhaps one of you could go over the life cycle of malaria, because that's the subject of this paper, malaria parasites. Maybe who wants to do that? Tell us, starting with a mosquito biting someone.
I'll do a little bit.
Go ahead.
All right, so I will jump. Hey, are you? I think you might be muted there, Christina, if you were trying to jump in.
Yes, I was. Sorry, I was
there.
distracted by some crashing in the kitchen. I said I could also contribute. I've actually managed, this is amazing, I managed to copy the life cycle into the paper just so I can remind myself.
Did?
Yes, I don't know how I did it, but it worked well. So
I'll start off with the first part with the sporozoites, and then once we get to the liver. So, well, now that she's got it in front of her, she's going to do it even better. But, this is the big thing that we've talked about several times, which I think is really interesting and important for our listeners to understand when it comes to vaccines, is so the female Anopheles at night, in the dark, interesting enough in most cases, will actually insert the proboscis. And there's actually a probing, and it's a heat-based probing, really actually trying to identify a blood vessel, a capillary. And then they will actually inject the sporozoites. And within minutes, and this is really critical, those sporozoites that some of our vaccines target will actually end up making it to the liver. In the liver, there's a maturation that happens that then leads to, and Christina, I'll let you jump in.
Yeah, so within the liver cells, schizones develop, which then rupture out of the liver cells, and then they start infecting red blood cells. And so that's the kind of erythrocytic cycle that we're now talking about. And within the erythrocytes, the parasites develop into from immature trophozoites, either into, so they're maturing, and eventually schizones develop again, just like they did in the liver, and they will rupture and they will reinfect other red blood cells. So that cycle continues, and that's what ultimately will give rise to the kind of fever patterns that we see in different malarias. In other other trophozoites, they will continue to develop into gametocytes. And these gametocytes, when they are taken up by the next blood meal, by the mosquitoes, will be taken up. And they will mate. So we get female and male gametocytes, micro and macrogametocytes, although I might have got the order wrong, I'm not sure. And then it's actually the macrogametocyte that enters the microgamete that enters the microgametocyte. So it's quite complicated. I suppose that's a detail that we don't need to go into too much. Sorry. So these then develop into eukinates, and these then develop further into oocysts. And within the oocysts, we have sporozoites developing. So the oocyst eventually ruptures, and these sporozoites, they get into the salivary system, and then we're back at where the mosquito does her probing action in the dark on a human and injects more sporozoites into the liver. And I think that's the cycle closed. So this paper exclusively focuses on what's happening in the mosquito.
Right. As you say, when the mosquito is injecting, it wants a blood meal, but it first injects some anticoagulants and painkillers so you don't slap it away.
A pharmacopoeia.
Pharmacopoeia, right. And then it starts to suck the blood out. And I have a wonderful movie I found years ago of when the mosquito starts to suck, the capillary collapses. It's sucking so hard and it's pulling all the blood out.
Oh, wow, that's amazing. You should share that.
So I will. It's really beautiful. It's a probing. It's looking for a blood vessel, and it finds one. It puts it right in. It's amazing that it can do that. And then it starts to suck. It puts in a little bit of the anticoagulant and the pharmacopoeia, and then it starts to pull out the blood. And of course, the deposition is where the sporozoites come out and go into you, and then you're screwed.
And it is amazing. It's just minutes. This window is minutes that you've got to get these before they end up in the liver.
All right, so let's go through the experiments. Very nice series of experiments. So
beautiful
first of all,
figures, I have to say.
In Plasmodium, all the Plasmodium genomes, there's a single gene encoding this protein QC. The gene's called QPCT, and they have different sizes. They encode different proteins, different percent identities. The Plasmodium QC has a transmembrane domain, and so it's stuck on the membrane of the cell, and they can make some of these structures have been solved, and they have built some homology models of these proteins, which are really amazing. They have 5 conserved blades of anti-parallel beta sheets. They kind of look like fans there. They're really beautiful.
Yeah, they really, the figure 1 people should go to and look at, A, particularly in this case, but also, B, D, and, well, C's not as exciting, but D is beautiful.
They make recombinant protein, and they show that it is catalytically active. It has cyclase activity. It will make a cyclic form of the glutamate. And then if you change 2 amino acids in the catalytic site, so you can see on the protein where the catalytic site is of this enzyme where the activity occurs. You can change 2 amino acids there. Now the enzyme is no longer catalytically active, so proving that this making of a cyclic glutamate is dependent on this enzyme activity. Now they look at the transcriptome data that have been published. In other words, people extract RNAs from various of these forms that we've just talked about of the parasite, gametocytes, ookinetes, oocysts, sporozoites, and they can see where the RNA is produced and then by extension where the protein might be produced. And you can see the QC is in the RNAs in gametocytes, ookinetes, oocysts, sporozoites, and these are all stages important for disease transmission, so that's quite interesting. The protein's not been found. The protein has not been found in asexual blood stage proteomes, but they find increased transcripts in female gametocytes and sporozoites, in Plasmodium berghei. So that was Yoli. How do you say that? Yoli? Plasmodium Yoli?
I'm not sure.
Now, let's say Yoli.
Where are we right now?
Dixon is published expression data, Plasmodium Yoli or Yoli.
Okay, let me find this and I'll try to.
And then
I think so. Think of their Latin, so UL-E.
And then Plasmodium berghei, they look at it's also observed in gametocytes, zygotes, ookinetes, and sporozoites. So all these expression data show that or suggest that this QC, where it's produced, where it's expressed as RNA, are in stages responsible for transmission. And they confirm that by they make a transgenic parasite that makes a c-Myc-tagged QC. So they put a epitope onto the QC, which they can then stain with antibodies, and they put that in the P. berghei, and they confirm protein expression in all these stages where they've seen it by transcripts. So bottom line, looks like this is made in the stages that are important for transmission. So then what do you do next? You want to know what this protein does? You knock it out. You take the gene out, and they do both P. berghei, which is a rodent plasmodium, and they could use a mouse model, and then the human parasite P. falciparum, and they delete the gene using standard techniques. These altered parasites lacking the QC gene, they have wild-type growth rate, asexual growth rate, they have wild-type gametocyte production, they have wild-type numbers of mature O kinates in vitro, but when fed to Anopheles stevensii, these null parasites make similar numbers of oocysts as wild-type parasites, but when they look at them closely under the light microscope, they see these aberrant, dark-colored, mature oocysts in maybe 65% of the null infected mosquitoes. So mosquitoes infected with parasites lacking the gene, the QC gene, they're dark-colored. And they don't see this in wild-type infected mosquitoes. They don't see these dark-colored O. kinates oocysts before day 10 post-infection, but they do see them either inside or during release from oocysts between day 14 and day 21. And what are these dark-colored oocysts and sporozoites? They are coated with melanin, a dark, insoluble pigment. And this is a known mosquito anti-plasmodium immune effector response to put melanin on proteins, and it hasn't been seen before in P. berghei, but it's been seen in some other systems. So day 21 post-infection, again with these null parasites, they can see non-motile enlarged sporozoites covered by melanin in the hemocoel of the mosquito. It's the circulatory system of the mosquito, essentially.
Yeah, it has this hemocoel.
Often in clusters, often attached to salivary glands. And at this point, the number of null salivary gland sporozoites are reduced compared with wild type. But most of the sporozoites within salivary glands had a wild type morphology, long and slender, without any signs of melanization. And they say this suggests that the sporozoites escape the mosquito melanization response once they invade the salivary glands. So they seem to be melanized in the hemocoel, and once they get into the salivary glands, that doesn't happen any longer. So they do some scanning electron microscopy of these oocysts and sporozoites from the null parasites, and they can see sulfur in the dark-colored parasites, which is indicative of pheomelanin, a constituent of melanized O. kinates. And this dark-colored material, which they call melanin, mainly located inside ruptured oocysts and on the surface of sporozoites. These are, by the way, as motile as wild-type sporozoites, so the motility is not decreased. So this just shows that the viability of the sporozoites that invade the salivary glands, they are still viable, and the reduced numbers likely result from immune recognition and melanization of sporozoites while they're on the way to the salivary gland in the hemocoel. So again, you have you take out this QC gene, and the oocysts and the sporozoites get melanized when they're in the hemocoel. And if you introduce the gene back into P. berghei or P. falciparum, it now complements the defect, and they no longer get melanized. They have wild-type like oocyst and sporozoite development without signs of melanization. Now, why is that important? Well, when you knock out a gene, it's a genetic manipulation. You could be accidentally making something else altered.
Mm-hmm.
So you put the gene back in to make sure that that's not the case. And if putting the gene in now corrects the defect, then you know that what you're seeing is due to the original gene deletion. It's called marker rescue. It's an old name called marker rescue. Most people wouldn't know that name any longer. All right. So then they make, this is very cool, they make a P. burgeri mutant with a QC with 2 amino acid changes in the catalytic site, those same 2 changes that knock out its cyclase activity. What do you think's going to happen with that one? It gets melanized, right? It's this enzyme, so that shows that the enzymatic activity of this QC protein is important to prevent melanization. So let's make sure everyone has got this so far. So we have this enzyme in Plasmodium, QC. If you knock it out or if you make it catalytically inactive so that it can't cyclize a glutamine or glutamate, the parasites get coated with this dark pigment, melanin, and it's not good for them. It seems to impair them. So then they ask, they do the same experiments in P. falciparum. All of these so far have been done with P. burgeri. They make a QC null P. falciparum. They get melanized oocysts, and again, so in both P. burgeri and P. falciparum, without QC, you get melanization of oocysts and sporozoites of both rodent and human malaria parasites, so it's conserved. All right. I'm inheriting Daniel's all right. Look at that. It's not good. All right, so what is causing this melanization? How are the parasites melanized? Wow, it's a tough word.
Melanized.
Melanized. So they want to know if just being dead triggers melanization. All right. So in other words, if abnormal or dead parasites, even if they have QC, would that trigger melanization? So they look at oocysts of 4 published P. burgeri mutants, and even with the defects that are seen, they have defects in sporozoite formation, or egress from oocysts, or invasion of salivary glands. Despite these defects, they don't see melanization. So making aberrant parasites is not a trigger of melanization. And then they ask, does melanization, with the elimination of these sporozoites, so remember, QC null sporozoites get melanized and then they're eliminated. What's eliminated? Because the numbers are lower in those. What's eliminating them? They say, well, maybe hemocytes, which are kind of like the macrophages of mosquitoes, right? Hemocytes, they can disrupt hemocyte function by injecting the mosquitoes with polystyrene beads, which I guess trash the hemocytes.
They're kind of getting all full and busy with those. So
with those,
not gonna function.
And then they ask, if you disrupt the hemocyte function, what happens to the sporozoites? There's no effect on wild type infected mosquitoes, but the number of QC null sporozoites go up, because remember, QC null are going to be melanized, and if they're melanized, they're apparently destroyed by hemocytes. So if
going to be phagocytized and then destroyed. Yeah.
So that if you eliminate hemocyte function, the numbers of melanized sporozoites go up. They also disrupt the complement system. They have a serine protease that they can inhibit to disrupt. That has no effect. I'm sorry. They disrupt it with an inhibitor called LRIM1. You don't need to know that. It's an inhibitor of the complement system. That doesn't have any effect.
That won't be on the test.
No, don't worry about it. We're not going to ask you that. Just say that disrupting the complement system doesn't have any effect on the number of QC null sporozoites. Then they have there's a serine protease, which apparently is a critical activator of melanization. They can inhibit that serine protease, and that reduces the percentage of null infected mosquitoes with melanized oocysts. So it makes sense. You're inhibiting the melanization enzyme in the mosquito, so you get fewer oocysts that are melanized. So all of this tells us that sporozoites without this QC enzyme are detected as non-self, they call it, and eliminated through melanization. I wonder if it's the melanization that makes it non-self, though.
I'm sort of thinking that the ability to not get melanized is what prevents them from, in the first place, being recognized. And here you've taken away that ability. They can't do this cyclic conversion. So now they get coated in melanin. It's almost like the melanin is an opsonization. Now they actually get pulled by the phagocytes.
So having the cyclase, if you're not cyclized on glutamines or glutamates,
if you can't make the glutamine cyclic, you can't protect yourself from the
mosquito recognizes it as
melanin
foreign.
Binds it. Now it's been tagged as non-self.
So the melanin is the secondary effect of being recognized as foreign. And then if the Plasmodium has the QC enzyme, then it will block these glutamines, and the mosquito won't be able to recognize.
It'll pass right through its
it's not recognized as non-self.
Yeah, and the heme, it's sort of tough, right? Because we're doing like mosquito immunology here, but mosquitoes have this sort of open chamber, right? They don't have a separate lymphatic and a separate circulatory. They have this hemocoel, C-O-E-L, where there's the hemolymph, and then they have different immune mechanisms. But the mosquito has to protect itself, or ideally is able to protect itself, recognizes non-self, tags it with the melanin, gets taken up by the hemocytes, their phagocytes.
Right.
But
the insect immune system is pretty cool, isn't it?
I like it. That's why I like this. It's a little mosquito immunology.
I was going to do my first postdoc was going to be on Drosophila immunity. But then, I met my leishmaniac husband, and the rest is history.
Well, I think it's, a lot of times when people do the malaria life cycle, they only do in humans. They forget about the whole
mosquito part.
The mosquito part.
All right. So now they want to address this issue of why they don't see melanized oocysts, even in the QC null Plasmodium, before day 10. And they're saying maybe if the oocysts are still inside the sporozoites are still inside the oocyst, or they're protected in some way. So they delete genes involved in sporozoite formation or in sporozoite egress. And so then they have now double knockout, which means they're deleted in those genes as well as the QC gene. They make wild-type numbers of oocysts, but the melanized oocysts are completely absent. So if you inhibit sporozoite formation or egress, you now don't get melanization. And in contrast, you get 80% of mosquitoes have melanized oocysts in mosquitoes, not only lacking QC, but the trap gene, where the sporozoites are released into the hemocoel, but they can't invade the salivary glands. So there you get melanization when they're trapped in the hemocoel. So they conclude that melanization only happens when the oocysts rupture and the sporozoites come in contact with the hemolymph, come out and into the. And then once they go in the salivary glands, they're protected.
It's too late. Yeah,
it's too late.
They're out of that immune environment.
That's interesting, right? And they have the genetics to do that. That's pretty cool. They can mess with sporozoite formation or egress. They can mess with
sporozoite
invasion
release
of the salivary glands, right?
Yeah.
So if you don't get in the salivary gland, you get a lot of melanization. They can't be protected. It's very good. Good system. All right. And finally, circumsporozoite protein, a very abundant protein on the sporozoite surface, a target for vaccine development for many years, right? That's what Victor and Ruth Nussenzweig worked on for years.
Yeah.
So they thought maybe this is a target for immune recognition in these null parasites. So they looked at the sequences of of these protein of circumsporozoite protein, and they see multiple peptides with P-glu at the N terminus, this cyclized form of glutamate, there's a 5 amino acid region that has. Then there's a 5 amino acid region that has a proteolytic cleavage site, and they think that cleavage there gives you a new glutamine at the N terminus, and then that can be modified to be cyclized.
Or not with the nulls.
Or not with the null, right? So they make cell lines that make a P. Bergii cell lines that make an altered CSP protein, circumsporozoite protein, where they replace glutamine with an alanine that would prevent cleavage and formation of the P-glu.
So you won't get this cyclic.
You won't get the cyclic thing, and then they show that this doesn't get cleaved. It does get cleaved. I'm sorry, it's just replacing that amino acid. So you're replacing the glutamine with an alanine. So now you're not going to have a glutamine there to be modified.
You get cleavage, but an alanine, not a glutamine that can be.
Right. And those oocysts are just like the QC null, no melanization in the first 10 days, and then day 14 to 16, you get abundant melanization. So QC is still there. You've now removed the glutamine, which is the target, so the protein can't be cyclized, and it doesn't get it gets melanized because it can't be hidden from the mosquito immune system, right? So a single glutamine residue in the circumsporozoite protein triggers immune recognition, and that causes melanization. And the parasites evade melanization, immune recognition, and melanization by making this enzyme QC. It modifies the circumsporozoite protein, and now it is not recognized as self. So what they have shown is that there is an actual target of the QC enzyme here in the circumsporozoite protein. They also say there are probably other targets as well.
Yeah, I mean, it's beautiful the way they pull it all together.
It's very nice. And so it sounds like this QC could be a drug target, right?
I'm trying to figure that out. I was thinking about that because they do start off by letting us know how ubiquitous this is. And so
it's in mammals too, right?
Yeah. So I'm trying to figure, is it essential? Because do you want to do this somehow in the mosquitoes? Like, spraying instead of to kill the mosquitoes to somehow allow them to eliminate the malaria parasites, right? Because this is all in mosquitoes. You have to treat the mosquitoes with something.
It's not going to help to treat people, right?
Right, because this is all maturation going on in the hemocoel of a mosquito. So, but you still care about mammalian toxicities because if you're treating the mosquitoes and it gets into the water, gets into the environment, I'm still trying to figure out where you put this in as a therapeutic without safety
Maybe
issues.
It would be a genetic approach that you'd have to take.
Could release genetically modified mosquitoes.
I haven't thought it through. I'm just kind of throwing it out there.
It could, because they do that with Wolbachia, right? They release Wolbachia modified mosquitoes for dengue. So in people, we have QCs, and but is it possible that in people that these modified glutamines would otherwise be recognized as foreign and melanized by the mammalian QC?
So we don't do this melanization thing.
We don't do that. So QCs have other functions.
Yeah, we have other functions. And then the issue is how similar is a parasitic QC enzyme to a mammalian enzyme or a bird or
Yeah, apparently they're very different. So the mammal and the others, all the others are different.
If they're
classes.
diverged enough that the active sites are different enough, because that's what I worry. You always worry about that. Okay, we're targeting this insect, QC, but are we going to somehow interfere? Like, is the catalytic site, the enzymatic site, similar enough that small molecule is going to have some off-target issues.
It's a weird thing that a glutamine, an N-terminal glutamate or glutamine, is going to trigger recognition.
Yeah, it is interesting. Yeah, and somehow by giving it a cyclic form, you basically prevent that trigger.
But there must be a lot of proteins in the parasite that have N-terminal glutamines, right?
Oh, yes.
Besides circumsporozoid protein, right?
Yeah, but the circumsporoid protein is really an immune target because it's covering the surface, right? So it's
that's true.
Yeah,
so all right, I guess if you look in the mosquito proteome, then there must be no N-terminal glutamates. Otherwise, they would be subject to this recognition, right?
Yep. Yeah, I mean, they do talk, and I'm not sure, like, if I understand how they're going to take advantage of this, but not only do we have QC genes, but a lot of parasites do, and they bring up toxoplasmosis gondii, right?
That's
which
right.
We just talked about. But I'm trying to figure out how do you target it? How do you sort of drug this in a safe, effective way without off-target concerns?
Yeah, they don't. They say it could be a drug target, but they don't go into it.
Yeah, I don't see. Yeah, I had run across this paper before and spent some time. It's a beautiful paper. But I always try to bring things back. Maybe it's the physician part of the scientist. Like, this is great. It's fascinating. It's a beautiful paper. Hey, by the way, can I do anything with this?
What is interesting to me is that the mosquitoes do want to counter the parasite, right? It's not that they say, go ahead, reproduce all you want.
Yeah. So the mosquitoes do have an immune system, and it is targeting, and to the point where the parasites had to actually evolve to evade that immune system.
Would the parasite kill the mosquito if this didn't happen?
There is a certain issue, right? I mean, so they're evading it right now with this mechanism, but there is a certain issue where if mosquitoes have too much of a parasite burden, it can have a negative cost.
So it impacts their fitness, and then they don't do well. Yeah, it's interesting. I never thought of Plasmodium impacting a mosquito. I mean, most of us don't care about mosquitoes, but in the biology world, it's interesting to know. It's just like many viruses don't impact the health of bats, right?
Yeah,
it's the same idea here.
Probably true.
I always assumed that the Plasmodium didn't impact mosquito health, but as you say, it does. So that's why they're making an immune response.
They're worried about the mosquitoes? Is that what it's?
Well, no. I mean, the mosquitoes are an entity that have evolved to survive, right? And if getting rid of Plasmodium is one of the survival mechanisms, that would be evolutionarily selected for. So
yeah, no, I mean, if there was a way to create a mosquito that could not allow this cycle, the sexual cycle of Plasmodium, that would be fantastic. If that then became, the mosquito, maybe we could also convince the mosquito, now that we made them better, they wouldn't, bite us anymore.
How would you make a mosquito resistant to infection with the sporozoites, right? Is that the stage that's going in them? Remind me.
No, so they're ingesting the gametocytes. And then ultimately it's the sporozoite, which comes to us, but it undergoes that stage, which I think we went through, where you end up with the oocyst. And then, but so it's when it's in the hemolymph, the hemocoel, when it's the most vulnerable. So if there was a way to modify a mosquito to be able to pick it up despite this attempt at immune evasion, that would be ideal.
Or maybe even preventing the sexual, the mating essentially between the micro and the macro gametocytes.
Yeah. Yeah. Very interesting.
Yeah, it's fascinating.
It was a beautiful paper as well, and really well written, and the figures are just beautiful.
All right, that brings us to our new case. What do you got for us, Daniel?
All right. I have to say, I was tossing over in my head. I had, I had a couple really good cases that I was, that were vying for a spot here. So let me go, because I think I pasted in my new case, or at least some notes for my new case. Let me get to that section. All right. Okay. So this is I'm going to claim this is a fun twist. Our emailers can tell me if I survived, if that's true. But I was recently chatting with Vincent about. I went to a gala, a dinner, a fundraiser in Central Park for the Wildlife Conservation Society. And apparently, Jeff Bezos was supposed to show up, but he just didn't even call. Didn't say anything, just didn't show up. There was an empty seat. I think he was on his yacht in the Mediterranean. Chevy Chase did show up, so that's okay. But more importantly, Paul Cayley was there, so the head vet for the zoos. But it got me thinking, and I will present the case here. That's a clue, by the way, of a 19-month-old female who seemed to be having some issues. I was taking care of this 19-month-old female. And I noticed that she really had this significant increased respiratory effort, even with minimal exertion. I was kind of wondering, maybe there's something going on here. Me, I'm listening to the heart and lungs. Everything seems fine. I'm reviewing a little bit of the past history. Have there been any medical problems going on here? Now, I do notice some loose stools. And then there is some facial swelling, which actually got quite significant. As I'll mention, no past medical, no past surgical. Fairly confident that there's no history of toxic habits, no smoking or drinking of alcohol, certainly not HIV positive. And as I mentioned, the only thing on exam was this increased respiratory effort. It was actually quite audible and made me quite concerned. This prompted some testing. And I will say we ended up with a couple positive tests. We got a positive stool test for canine coccidia and a positive stool test for Giardia. I'm not sure if it was Lamblia duodenalis or maybe, and I suspect, another species of Giardia. So for our listeners, what is going on here? I have a 22-year-old daughter in the home who had lots of exposure to this 19-month-old female. Should I be worried about my 22-year-old daughter? And I'll leave it there for some investigations. I'm not sure what ChatGPT will do with this.
19-month-old female. She hasn't gone anywhere, right?
She's been around a lot of other, young
other
females,
but she hasn't traveled.
So no travel. This 19-month-old female has been, in the local region. No overseas travel.
Can I just clarify who treated?
She has not eaten at any restaurants.
I was just going to clarify who was treating. Was it Paul Cayley who was treating, or was it you who was treating?
No. I was actually taking care of this 19-month-old female. I was just chatting with Paul Cayley. Actually, just got me thinking it would be fun.
Yeah, I was just wondering because he mentioned he was the head veterinarian of the zoo. So I thought, maybe.
Should we go back and listen to the Paul Cayley episode?
We should put in a link to the Paul Cayley episode. Paul and I were chatting about that. That was fun for our listeners who may not have listened or watched. That was actually a video episode as well.
It was really good. We went to the Bronx Zoo for the reveal, right? So do you think that's relevant to this case, Daniel?
I think there's a hint there.
Wow, that's curiouser, curiouser and curiouser. All right, so the 19-month-old female is someone's kid and is brought up in that home. Who else lives in that home? No?
So I didn't say this was a goat. I never said it was a kid.
You never said it was you didn't even say it was a human. Look at that, 19-month-old female.
There you go.
Has been in the care of several people, including your daughter, right?
Yeah, I was thinking that canine coccidia and the Giardia, possibly subspecies Canis, might be a bit of a hint. But we'll leave that open for our guests.
This is very interesting. Well, I don't need to ask anything more. That's great.
This is going to be fun.
I like that very much. We have a couple of emails that have been sitting here for a long time. Do you think we could work our way through them?
Yeah, let's hit a few.
There's just three, and we could do all three and be done with them. Christina, can you take that?
Obada writes, "Hi Vincent, I'm a regular listener to TWiP and really love the way you explain things clearly. Anyway, the reason I'm contacting you is because our family has recently got scabies and it took us 5 long months to get to the right diagnosis. I feel this is a disease that both parents and physicians should be educated on. The main issue was every doctor seemed to confuse it with allergy and eczema, and even when I suggest the scabies, our after Googling our symptoms, the doctors always look between the finger and wrist and rules it out. Well, in our particular case, there was nothing between the fingers until 5 months in, and even then it was only 1 out of 4 infected family members that displayed this. It was only after I got me a cheap Amazon USB microscope and my wife, non-medical, mastered the art of skin scraping that we got photo and video evidence to show the doctors so they won't go back to eczema crap. That is pretty amazing. Yeah, I like that. Lots of initiative there. By the way, during that last month where some doctors were saying it might be scabies, but no one here in Ontario, Canada would dare to prescribe ivermectin in addition to the permethrin cream. I guess the whole ivermectin COVID crap harmed the wrong people. I wasn't sure whether I had to bleep that out.
No, I think CRAP is a French word. So.
Okay, we did 2 applications of permethrin cream, all of us 2 weeks apart, and the leaflet tells you that the itchiness could persist up to 6 weeks. So we went back to our normal lives, and then 4 weeks from the second application, we managed to see life might all over our bodies, and that's when we found a new family doctor and showed him the evidence that we got oral ivermectin and took it a week on top of yet another permethrin application. So far, we seem to be doing well and no sign of new mites. Still have a second dose of ivermectin 14 days from the first one and hoping for the best. Sorry for the rambling and ranting, and I appreciate you reading all that. Best regards, Obada. I thought if the whole family has got the itches, you would probably not. Your first thought would probably not be eczema, would it, Daniel?
I think you're overestimating my colleagues and I. And thank you. But no, unfortunately, I will often hear stories like this, and then finally we cross paths, and part of it I have to say is there has been a negative impact on the willingness to prescribe ivermectin when appropriate, because of all this concern that they're going to think I'm one of them. They're going to think I'm doing it in an inappropriate manner. But unfortunately, I think, and hopefully our show helps with this, the awareness that scabies is a thing, that scabies is something that's out there and needs to be recognized and treated. Hopefully we're getting the awareness.
I actually just remembered a funny story. So my son, James, whom you have met, Daniel, he had also rashes, and it just looked a bit weird. So I had taken him to the doctor, and they were really unable to make a diagnosis, and he had various treatments that made no difference. And it was essentially, in the end, it was just by sheer coincidence, he came along with me to the vet, and he was holding the cat, so the cat would hold still for her treatment, and the vet said, "Oh, James, you've got flea bites on your arms." And that was that. We then cleaned the house and treated the house, and we've never seen a flea bite since. So that was, I suppose, quite similar.
Fantastic.
Although not as distressing.
Yeah.
Daniel, next one, please.
All right. Jillian writes, hello. I just wanted to reach out as listening to the latest episode of TWiP. I noticed that my guest was skipped, and sadly, I realized that I'd accidentally switched case number 212 with 121. With that in mind, I wanted to quickly send along my response to the one sant question regarding the pronunciation of centimeter for your interest. I have to say that this was almost as interesting as a case study, as actually I'd never heard it pronounced as centimeter before. In fact, a previous episode had found me on Google searching for blemish-specific measurement that I was unaware of. In discussing with my friends, though, we suspect the pronunciation is connected not to Canadian English at large. What about colors, eh? But rather could suggest a Quebecois specific linkage. After all, cent is pronounced cent when it is 100 en français. It also seems like there might be some connection to French as being seen as a more elite language at some of the long-established medical schools. Hope you found that all interesting as I did. All the best, and I'm glad I guessed right on the case as well. Jillian?
Centimeter.
Do we all speak French?
You say centimeter.
Well, interesting. I say centimeter, and I used to say it was signaling that I had gone to NYU. So everyone who graduated from NYU medical school in my generation would say centimeter and umbilicus. But I met a recent grad, and apparently the old guard that used to pronounce things that way is long gone, and now people call it the belly button and say centimeter.
Centimeter. Umbilicus, huh?
I would have said umbilicus. Yeah.
Yes.
All right. Joshua writes, Dear Doctors Despommier, Griffin, Nowla, and Racaniello, I trust that you, your loved ones, and all my fellow listeners are enjoying the festive season. Nowadays, however, thanks to your addictive podcast, to which I have been glued during the Christmas holidays, I view the eggnog, the turkey, the ham, and even the plum pudding with some alarm. What pesky parasites perhaps proliferate? Oh, what a series of words. What pesky parasites perhaps proliferate?
Yep.
I like that. That's great.
Within these and other delicious delights, it is a warm 27°C this afternoon here in Launceston, Tasmania, on this, the last day of 2022. I must confess, I await 23 with trepidation bordering on dread after all the world has been through during the first 3 years of the 2020s. But on to the case. All right, so now he's looking at the case here. So I don't think
I guess this is an old case.
This is an old case. But there is some... Here, now for some peculiar Christmas presents, parasitic but not infectious for the TWiP team to enjoy. Okay. For Dr. Despommier, I will mention that Tasmania used to have a severe problem with hydatids caused by Echinococcus granulosus. In 1966, over half of adult sheep had hydatid cysts, and more than 1 in 8 rural dogs had adult worms. In the 1950s, the Tasmanian surgeons operated for human hydatid disease about once every week, and hydatids caused the death of a Tasmanian child every few years until 1968. However, government action commenced in 1965 testing and quarantining infected dogs and sheep, and the last new human case was diagnosed in 1976. Thanks to this eradication campaign, Tasmania was declared free of hydatids in 1966. Well, that's cool.
1996.
96, sorry. Next, I must thank Dr. Griffin for alerting me via a mention some years ago on TWiP 129 to a very rare endemic Tasmanian helminthic disease of which I had never heard, parasitic myositis caused by Haycocknema perplexum. Years ago, a visiting friend of my mother made the adventurous food choice of a rare possum steak that looked like quivering meat jelly. Hopefully, the marsupial meal did not conceal nematodes awaiting their own feast of fresh flesh. Well, this guy or woman loves to write alliterations. Doubtless, in his time in Colorado, he treated cases of Rocky Mountain spotted fever, but has he ever heard of its Tasmanian equivalent, Flinders Island spotted fever? It's caused by the bacteria Rickettsia honii, transmitted by tick bites. The ticks are parasitic on native reptiles.
I've got to go spend some time down there.
Tasmania?
I have been to Tasmania. It's absolutely beautiful. Yeah.
I was going for the parasites. You went for the view,
well,
obviously.
that was before I was a proper parasitologist. So it was quite a long time ago.
For Dr. Nowla, I will make reference to the well-known devil facial tumor disease, a transmissible cancer first identified in 1996 that has gravely reduced the numbers of the Tasmanian devils, Sarcophilus harrisi, leading to it being listed as an endangered species. It is the largest remaining carnivorous marsupial, as the Tasmanian tiger Thylacinus cynocephalus became extinct in 1936. On display in the local museum is a very sad silent movie of the last thylacine pacing in its cage at the old Hobart Zoo. At a local zoo, I have seen the Tasmanian devils being fed. They chomp through kangaroo bones like butter, and I hope they will not go the same way. I haven't yet listened to the discussion of this disease on TWiP 371, but will soon do so.
Daniel, do you remember the thylacine that we saw in the Hunterian Museum when you visited Glasgow? There was a stuffed one there, wasn't there?
Yes.
Yeah, it looked a bit sad, the poor thing.
Yeah, and I've seen that silent movie,
too,
actually.
Lastly, for Dr. Racaniello, the poliovirus expert, I will mention the Tasmanian infantile paralysis epidemic of 1937 to 38. This was the second worst per capita epidemic of polio ever recorded, only surpassed by the 1924 Icelandic outbreak, with a case rate of 421 cases per 100,000 people. And Kilalia, the Great Scourge, Sandy Bay, Tasmanian Historical Research Association, that's the book, I guess. What is now a local school for children with special needs was established in the aftermath of that epidemic as the home for crippled children. I know a retired nurse who was born 10 years later, but still remembers her mother being very anxious about letting her visit the neighborhood playground. Such was the fear, even in the 1950s, of catching poliomyelitis. Oh, I'd never heard of either outbreak. I have to go look that up. Thank you. That's very cool. It's not cool, but it's interesting. May I finally thank him for the fascinating interview on TWiP 962 with Dr. Chiara Mingarelli about black holes and related topics. My honors degree was in astronomy, and I have always retained my childhood fascination with outer space. Yours sincerely, Joshua. Okay.
That was lovely.
Thank you very much. That was very nice.
Thank you for the presents.
That is TWiP 218. You can find the show notes at microbe.tv/TWiP. Questions and comments, your case guests, go to TWiP at microbe.tv. If you like our work, consider supporting us, microbe.tv/contribute. Daniel Griffin is at Columbia University Irving Medical Center, parasiteswithoutborders.com. Thanks, Daniel. Thanks for coming.
Thank you.
Christina Naula is at the University of Glasgow. Thanks, Christina.
Oh, thank you. It was fun, as usual.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiP and Ronald Jenkins for the music. Whose turn is it?
You know, we should do it together, right? Because we're
here.
Yeah,
right?
you do it.
We'll actually get it all in sync, I think.
All right, we can try. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon.
Another TWiP is parasitic.
Boy.
That was good.